When a patient is prescribed a generic drug, the medical system operates on a straightforward assumption: a copy is a perfect copy. By definition, a generic medication like metformin is synthesized chemically in a lab to be atom for atom identical to the original brand name. Biologics operate in a completely different physical reality. These are massive, three-dimensional proteins cultivated inside living cells. Because of this complexity, regulators approve biosimilars under a standard of being highly similar, with no clinically meaningful differences. They do not classify them as identical. Living organisms are inherently variable, meaning minor structural alterations, such as how the protein folds or attaches to sugars, will naturally occur during production. Even the original brand name reference product drifts structurally over its commercial lifespan. Creating a mathematically exact replica is impossible for the originator, let alone a competitor. In biologic manufacturing, the process is the product. The specific environment dictating how the protein is grown defines its final clinical profile. Passive safety monitoring systems, designed for stable, predictable chemical generics, lack the granularity required when the medicine in question cannot be perfectly replicated. Treating a biosimilar exactly like a generic introduces a gap in post-market surveillance. Monitoring these drugs requires a specialized approach that accounts for their biological nature. Biological medicines are not synthesized, they are grown. The production line is a living system, typically a cultivated cell line interacting in a turbulent bioreactor. A competitor engineering a biosimilar must develop their own proprietary cell lines and calibrate their own fermentation conditions from scratch. These independent biological environments directly influence how the cell secretes the target protein, guaranteeing slight variations from the reference product. As the cell constructs the protein, environmental conditions alter how sugar molecules attach, a process known as glycosylation. Then, specific scale-up and purification steps determine its final state, including its tendency to aggregate into clumps or retain impurities. Early biologic manufacturing relied heavily on intricate, manual purification methods that carried inherent risks for severe batch-to-batch variability. Today's analytical testing is highly advanced, but it still cannot map every single structural nuance that might affect a human patient. That inherent manufacturing drift means the laboratory cannot provide the final word on safety. The true clinical profile is only fully verified through the longitudinal exposure of millions of patients. The most critical safety threat facing biological medicines is immunogenicity. Because biologics are massive foreign proteins, they have the inherent potential to provoke an unwanted response from a patient's own immune system. The definitive case study proving this spread occurred in the early 2000s with the reference biologic epoetin alpha, marketed as Eprex. The manufacturer introduced two seemingly minor adjustments: a new rubber stopper for the syringe and the replacement of a human serum stabilizer with polysorbate 80. Inside the syringe, adjustments caused therapeutic proteins to aggregate. When injected, the immune system identified these clumps as a threat, generating neutralizing antibodies. These antibodies neutralized the medication and attacked the body's natural proteins, shutting down red blood cell production. The result was pure red cell aplasia. Patients with chronic kidney disease suddenly lost the ability to produce red blood cells entirely. Pre-approval clinical trials for biosimilars are intentionally abbreviated to prove comparability, making them statistically too small and too short to detect rare, delayed immunogenic reactions of this scale. The Eprex outbreak proved that seemingly trivial manufacturing changes can be devastating. Continuous, long-term pharmacovigilance remains the only reliable defense against unforeseen immune responses. When a doctor reports a standard adverse drug event, they typically record the international non-proprietary name, which identifies the active ingredient. Applying this standard to biologics breaks down. Multiple competing biosimilars share the exact same non-proprietary name. If a safety signal emerges, data becomes difficult to parse. Regulators cannot determine if the danger is class-wide or isolated to a specific batch. To trace these signals, the U.S. Food and Drug Administration requires unique four-letter suffixes attached to the non-proprietary name to force product differentiation. The European Medicines Agency takes a different route. They maintain the shared name, but strictly mandate capturing the specific brand name and batch documentation. The baseline requirement for accurate biological surveillance is universal: Effective signal detection relies on having the precise brand name and the specific lot number. Tracking this data is uniquely difficult when healthcare systems routinely mandate switching or substituting biological products at the pharmacy level. Patients transition to a new brand often experience the nocebo effect, reporting new or worsening adverse events driven entirely by anxiety about the switch, rather than any actual pharmacological change. Distinguishing a psychological nocebo reaction from a genuine manufacturing defect relies completely on absolute, batch-level traceability. Without it, safety teams cannot accurately monitor the biological supply chain. Because biosimilars rely on abbreviated clinical development, the burden of proving long-term safety permanently shifts to the post-market environment. Before a biosimilar even launches, manufacturers must establish a risk management plan to actively hunt for safety data missing from the initial trials. This requires dedicated, continuous immunogenicity monitoring, utilizing standardized diagnostic assays that can detect antibody formation over months or years of treatment. Surveillance must also look beyond the pharmacology. Analysts evaluate supply chain integrity, tracking cold chain failures and specialized injection device malfunctions that can degrade the biologic before it reaches the patient. To capture this complex, longitudinal data, pharmacovigilance teams pull real-world evidence directly from electronic health records, insurance claims, and specialized patient registries. Global coordination relies on standard-setting bodies like the World Health Organization to implement these rigorous reporting frameworks, particularly in emerging markets where biosimilar adoption is rapidly accelerating. The entire infrastructure depends on frontline data collection. Clinical prescribers and pharmacists are responsible for meticulous batch number documentation from the first prescription. The global biosimilar market is projected to save healthcare systems tens of billions of dollars, but those economics rely entirely on patients and providers trusting the safety profile. Pharmacovigilance for biologics is an active, rigorous scientific discipline. It is the mandatory infrastructure required to deliver the next generation of complex medicine safely.